started. Well, thanks for coming. Dr. John Field from the Northwest Fisheries Science Centers. Speaker today. Oh, sorry. Ooh, that's really first, doesn't it?
I got it though.
Yeah. Well, I was thinking, because it's groundfish you work on, you were the Northwest Center, but there's a small contingent of rebels from the Southwest Fisheries Science Center that work on the California and Northwest groundfish. And most of you know John, but if you didn't know that, he's really one of the up-and-comers. Actually, that's no longer true. You're not up-and-coming, you're there now. One of the experts in the field of fisheries management and fisheries science, and it's kind of follows along our talk we had, our seminar speaker we had last fall, Jason Cope from Seattle came down. Jason was talking about stock assessments and data for fisheries management. And John's going to speak today about ecosystem-based management, something that people have talked about for a long time, and how variation in rockfish recruitment fits into that. And as I said, he's one of the leaders in the world in this, and I'm not surprised, because I asked him for a little note to help me introduce him. He said he grew up in Santa Cruz, a mile from the lab up there. And then he said he did preschool through UCSC before moving on to graduate school. And I thought, that's really amazing. You could get preschooling at UCSC and then go straight to graduate school. So
I'm spent in the same town.
So it tells you how kind of advanced he is. Were you 5 or 6 when you got out of grad school?
You still haven't learned how to write.
Anyway, John received his BA in biology and environmental studies from UC Santa Cruz in 1994, and then took a direction that a lot of people who come to our lab think about. He went into the school at the University of Washington to get a master's in marine affairs. A lot of people that come and talk to us are thinking about, should I come to Moss Landing to get into science, or should I go somewhere else to get a marine policy degree? So John took the route of the marine policy arena, and then served as a Sea Grant Naus Fellow in 1988, 1990. And after being in DC, he wisely came back into the science world and got his PhD at the University of Washington. And his dissertation was application of ecosystem-based fishery management approaches in the Northern California current. And I'd venture to say that in addition to all the work he does for NIMS now, he's been spending a lot of time thinking and studying ways to apply ecosystem-based management to the fisheries. He's the PI for the Juvenile Rockfish survey team, and he's going to talk to you about that. And also the team leader for the groundfish contingent is a small group of rebels who aren't in the Northwest Fisheries Science Center. So John spent a few years working on the plan development team for the Pacific Fishery Management Council's planning process to figure out how to accomplish ecosystem-based management. And after losing a few years of his lifespan in that process, they are now starting to move towards implementing ecosystem-based management. And as I said, John's been thinking about this and working on how to implement ecosystem-based management for a long time, and we're lucky to have him here today to talk about that.
Thank you, Rick. I think one of the things I really wanted to accomplish too is actually tell you about the rockfish survey in your backyard, because it did strike me that not a lot of people know what we do. The Santa Cruz lab was in Tiburon in San Francisco Bay until about 1999-2000 when they moved to the current location in Santa Cruz. And they started this survey actually in 1983, so it's been going over 30 years. And at that time, they formed a lot of times with the Romberg Tiburon Center and other people up there, and had a lot of interactions. A lot of graduate students would come and work at the Romberg Center and then work on the cruise and publish and get hired. T. Sakuma was a Romberg person and several others as well. And I think we kind of lost part of that connection when we moved to Santa Cruz. We actually still work with some Romberg people, and they still put people on the cruises, but it's not as not as much interaction as it used to be. And we've built up a little bit with Marinovic and some others at UCSC, but not a whole lot. So it struck me that it might be a good pitch to come down here and just show you some of the results we've got. I think since Steve Ralston retired a couple of years ago, he's actually been more productive than he had been in a long time. And since I took over as lead of the groundfish team, I've been less productive than I have been in a long time. So a lot of what I'm going to show you is some of the papers that he led, sort of tying up some of the stuff he'd been thinking about for decades, literally for decades. And a lot of that stuff is just coming out, or is in review right now. And then we've been working with Jared Santora, Bill Seiderman, a number of others from the Fairlawn Institute. Jared's actually now C-Star at UCSC. And Brian Wells and other people as part of this integrated ecosystem management effort and part of the ecosystem indicators development effort to do different things, create different products from the cruise. So really what I'm going to highlight is not a whole lot of my work, but a lot of work I've been involved with in the recent years that's been led by some of these other people. But one of the larger objectives then is to get some of you all interested in what we're doing, because we've got way more data than we could possibly analyze, and I'm sure there's one or two pieces in there somewhere for people who are out shopping or just need something to do on weekends. So I put a short list, and it's definitely not complete, of some of the people whose material I stole in order to show off to you today. And again, that's largely Steve and Keith, but also Brian Wells, Jared Santora, and Isaac Schroeder, who's with ERD, Environmental Research Division in Pacific Grove. Eric Bjergstad, who's with our division, but he's located in Humboldt State, and his student Catherine Crane. They've been doing some process work. Bill Seiderman at the Fairlawn Institute, formerly with PRBO. We've kind of started work with him, and there was awkward transitions, but we've continued to do some work with him in recent years, and actually have been really productive. And one of the main reasons, again, was Jared Santora, who was with Bill and is just really embraced the whole data set. And with Isaac's help, Isaac's a physical oceanographer, and you'll see some of their stuff towards the end. They've really done more with this data, I think, than we've been able to do. Waldo Marinovic at UCSC has been putting krill biologists on the cruise. They've been doing all kinds of stuff that I can't wait to see with respect to doing size frequencies, analysis of brooding, species composition, all kinds of other stuff with a lot of krill we catch. We think we do a pretty good job on enumerating krill. He's done great stuff with that, and I think we're just beginning to see the fruits of some of that work. And then Jason Phillips and Rick Broder at the Northwest Center. We're kind of in the last couple of years trying to make this really a coastwide survey and effort, and they're a big part of the work up in the north, although we haven't done a whole lot with respect to analyzing a lot of that data we've just been collecting. So the main objectives, the other reason I thought this would be a good talk to give is because I just kind of gave it. I gave a short version at Western Groundfish, and apologies already to those of you who are seeing this again. Hopefully there's a few details that you missed. But we also had a program review, a NOAA science program review in September, where we basically did a very rigorous external review of all the data collection programs that support stock assessments and research activities in the agency. And so I had to pull together all this material for the program review, and so I had it kind of handy. And so this is what they wanted to see, what are our main objectives, and I'm going to be recycling a lot of the kind of stuff. But the main things we do, the survey started out really as a stock assessment survey to develop indices of relative abundance of young and meter rockfish to inform stock assessments. The idea being these are long-lived, slow-growing species that have very infrequent recruitment events. If we know when those recruitment events are coming, we can manage the stock better because we know when there'll be a big influx of biomass to the population. And some are less slow-growing than others, and I'll talk about Boccaccios being that example. But along the way, of course, we want to understand the physical and biological ecosystem factors that lead to these strong or weak year classes, process studies, what drives recruitment, the holy grail of fishery science. And then, of course, especially in recent years, we've been wanting to understand more broadly the spatial and temporal variability in the whole entire forage base, because we sample a lot more than just juvenile rockfish, and try and understand how that relates to higher trophic level productivity, how it's driven by climate, those kinds of things. So this is a core area that's been sampled since 1983. We are here, so this is, I don't know if it's your front yard or your backyard, I don't know what you consider it, but this is, with one of our stations here in red is not very far away from us. here. We've been sampling inside and outside Monterey Bay, as well as the Davenport, Pescadero, Gulf and Fairlawns, and Point Reyes lines for 31 years, doing the trawls at night, doing CTD casts and marine mammal observations, seabird observations during the day, especially around the Farallon Islands, one of the biggest breeding colonies for seabirds south of Mendocino. For reasons I'll get into a little later, since 2004, we've actually been surveying a broader range on our cruise from Cape Mendocino down to the Mexican border. So we've got a series of stations in the Southern California Bight and north of us. And then the Northwest Center in the last couple years has had a series of stations. Before that, they had a cooperative arrangement with the Pacific Whiting Conservation Council, which is an industry group. They wanted to develop these pre-recruit indices of Pacific hake, which has also very variable recruitment, which leads to very dramatic changes in the availability of fish for the fishery. It's the largest fishery on the West Coast, mostly off Oregon and Washington. They catch 200 to 400,000 tons a year. So that's a big fishery. They had a lot of money, so they were interested in doing recruit indices for that fishery. So far, we haven't successfully actually done that. It might be a good thesis project to figure that out, because the industry really cares. I talk to the industry guys all the time. We've been trying, and we're still doing work on hake. Hake are tricky, so are rockfish for that matter, you'll see. Anyway, so over the last few years, and we're really actually trying to collect a very strong collection of data from these coast-wide assessments, partially to cover the whole coast, but also partially to figure out where it is we need to sample in order to do these indices right, and you'll see why shortly. Up until 2008, all our cruises were done on the David Starr Jordan, a NOAA ship that had been around for a very long time. It was a great ship. Starting in 2009, we were on a different ship every year, to the point where last year, 2013, we were back on the David Starr Jordan, which had been decommissioned as a NOAA ship, bought by a contractor, and renamed the Ocean Star. It was still nice to be back, but you know, and then now we're in this kind of like, well, if we're estimating vessel effects, is it the same ship? Is it a different ship? It's not fished the same way, it's not run the same way, but it is the same ship. So we haven't tackled that question yet. Next year was supposed to be on the Reuben Lasker, which is the newest NOAA ship that will be based in the Southwest Center. Every time I see the schedule, we're still there. No one's X'd us out yet, but when you're in the government, you're always very worried about someone just taking that ship time and putting it in their column. So far, we've still got it. We do all our trawling at night because the fish can avoid the net during the daytime. We fish at a 30-meter head rope depth, so we're fishing, 30 meters below the surface, 30 to 40 meters. We've done a set of depth stratified tows historically, and that seems to be where there's a pretty good concentration of rockfish. They seem evenly dispersed throughout the water column, but this does affect the sort of catchability of some of the other things that we work with, where that probably wouldn't be the optimal depth for, say, anchovies or sardines. Use a fine mesh liner. This is really kind of a glorified plankton net in some way. When we were dealing with sort of marine mammal mitigation, things like that, they were comparing our net to the kind of net the salmon folks use, which is a Nordic midwater trawl. It's got about 5 times the net opening, much broader mesh. It's fished at a faster pace. That's fished at about 3.5 knots. We're fishing at 2 knots. So we're really going kind of slow and kind of deep, and fishing more like a really big Isaac's Kids or something like that, rather than the kind of big sort of trawl net, surface trawl that salmon people or bottom trawl that groundfish people are used to thinking about. We catch lots of stuff. We count them, identify them, sort them, do most of this stuff at sea. We bring some of the stuff back to the lab. It's a big pile of mush, but some of it's very beautiful. That's a juvenile cow cod. They're the prettiest, but we don't catch too many of them. We actually caught more than we'd ever caught before last year. It's very exciting. Oh, there, I knew there'd be a PC to Apple issue in there somewhere, or maybe that's not from that. Anyway, ignore that little bit right there. So we're targeting this ontogenetic stage, the pelagic young of the year. They're 90 to 100 days old. We actually standardize the index to a 100-day index to kind of try and be evenly comparing things across years. And this is after we think most of the density-independent mortality has taken place. So early on, once they're extruded, the larval abundance is generally considered to be an indicator of spawning biomass. We've actually used it that way in a number of assessments based on Cal Coffee data. And we think it's about somewhere between 50 and 30 days are the primary factors that determine the relative year class strength. That's when fish either die or they don't die. And small differences in the rate at which they die or don't die will lead to the relative year class strength that you see. So we think that year class strength is primarily set by the time they're about 90, 100 days old in the water column. Then they settle out, and then they become adults, and then there's surveys for this. But there's some density-dependent mortality in there that we've at times tried to deal with
in developing the indices. That's probably a topic for another day. So in addition to the network, we're taking a CTD cast, especially all day long, but also at night with every tow and collecting salinity, temperature data, density data, things like that. We're taking acoustic measurements. I think since the mid-1990s, we've been running acoustics, and since the early 2000s, we've been analyzing this. We've been plotting out krill hotspots. We're starting to look at some of the other species, mesopelagics and things like that. So here's, say, the red parts are krill hotspots in the California current based on the climatology that Jared did. We opportunistically catch big things like Humboldt squid and cut out their stomachs and do horrible things to them. We'd have seabird and marine mammal people basically doing observations all day long during the transects. That's been happening since 1987, so there's a nice time series, I think something like 50,000 miles of bird and mammal observations in the various databases we've done there. And that's largely through partners like PRBO, Point Blue, and Fairlawn Institute. The 10 rockfish species that we see the most of are up here. Widow, yellowtail, chili pepper, square spot, short belly, and striped tail are not commercially important. The rest of the species are. Short belly is ecologically important. They're probably the most abundant species, are small midwater schooling species that are very important prey to seabirds, marine mammals, everything else that is larger than it, including boccaccio and chili pepper. Then we get brown, blue, boccaccio, and canary rockfish. So these make up about 95% of the total catch, the winter spawners. These 10 are probably the only 10, at least at this point, that have enough data for relative abundance indices. As we do more in the south and in the north, there's probably other species we'll start to get enough of, but for now, these are the species we focused on. These are what they look like when we get them. They don't look like mommy and daddy. They can be rather tricky to tell apart. Some are a little easier than others. Keith is really the master at this. He rarely has to do fin ray counts and fun things like that, but at times you do. Some of them can be pretty tricky to tell apart, especially the Sebastes, I think the yellowtail black and widow group. So these species represent the vast majority of rockfish, commercial and recreational rockfish landings in California over the last 5 decades or so. So here's a time series of the relative rockfish landings in California, peaking in the early mid-'80s, falling very dramatically here, and then of course we had the fishing disaster. A number of these species were declared overfished, southern and northern species. I didn't put the northern on here. I'm focusing just on the ones that we have in the survey, but 3 of those, widow, canary, and boccaccio, were overfished. Several of the others were overfished or close to it in retrospect, but stocks had sort of rebuilt by the time we actually did the assessment that they were never formally declared overfished. And this really, if you look at the trends for many of the other species in the California current rockfish, as well as things like lingcod and other things, a lot of it looks a lot like this. We just hammered stuff too hard in the 80s and 90s. We finally let up around 2000 and shut down the shelf, cut back on the fisheries. The fishermen themselves are in a heap of hurt right now because the catches are so low, but the stocks are rebuilding, and I think we're now at a point where we're transitioning towards a much more sustainable management regime, and everybody's happy with that. Early on, in terms of using the indices in the assessments, things look pretty good. If you looked at the sort of the age data estimates of recruits, young of year recruits from the assessment, no emphasis here means that there was no emphasis put on the YOI index data. Basically, the midwater trawl survey was catching the big year classes and catching the small year classes. Boccaccio, very variable recruitment, and the indices seem to be catching that. Also seem to be doing pretty well with widow rockfish. For both these, during strong El Nino events, recruitment was crappy. So over time, we provided indices for 8 species over time. Prior to 1998, everything looked pretty good. In terms of why it matters, again, trying to understand, and boccaccio are the example of the long-lived, slow-growing. They're long-lived, but they're very fast-growing. The interesting thing about boccaccio is when you get a strong year class, and recruitment varies by several orders of magnitude from year to year, they enter the fishery very fast. So here's, this is boccaccio recruits here, and these are different catch rate time series, the orange and the purple, late and early, because you can't really compare the two. And then the percent of boccaccio is the total recreational catch. So what you see is that when you get a strong recruitment, you get a lot of boccaccio, high boccaccio catch rates, and a lot of boccaccio in the catch. Up to 35% of the catch was boccaccio back in the 1970s. This is just Southern California where most of the boccaccio are. So again, a strong year class in '84, the catch rates of fraction of boccaccio goes up. Same thing happened in '99. The trick is that in 1999, we thought boccaccio were tanking. And an assessment done in 2001, I think, estimated that it would take several hundred years to rebuild, that the only sustainable catch was 10 to 20 tons, and they basically shut down the entire coast in 2002 to rebuild Boccaccio, because what happened was the fishermen were saying, there's tons of Boccaccio out there, we can't get out of them, we're catching them left and right. A strong year class had come in '99, we didn't have the data in the assessment, which takes several years for those fish to show up in the fishery, so we didn't know a strong year class was coming. It came, the fishermen couldn't avoid them, and as a result, the entire fishery along the coast was essentially shut down, with the fishermen saying, you guys are nuts, there's Boccaccio everywhere, you don't know crap. So, you know, part of the problem was, well, we didn't see it in our index, so we were quite worried. And here was an example of where it would have been really nice to see it in the index, because we would have known that that strong year class was coming. You know, people were thinking of Boccaccio as being this thing, but really what it was is these are one-year-old fish that we caught off Santa Barbara. This is a 2010 year class that we caught in 2011. And when you get these strong year classes, they're just everywhere. Wherever you go, especially in Southern California, there's little Boccaccio everywhere. Even Milton talked about it in his book. He was on Cayucos Pier. He didn't get to catch him because his pole fell through the slats of the pier. And he saw it disappear into the water. Everybody else was catching Boccaccio. They caught 500,000 Boccaccio that year. It was a really strong year class, and he didn't get any. He's still very traumatized about that. To Milton. Or his pole. Okay, so and when we're doing the indices, we standardize the abundance to a common ontogenetic stage, 100 days, based on some simple math, basically accounting for the mortality rate and the growth of the different fish. What we do see, though, is that in years of high abundance, the fish tend to be larger. So we looked at the sensitivity to some of these assumptions, especially the natural mortality rate. It's relatively insensitive to that, and the rate's actually based on a little bit of data that is in itself another story. And so far we think it's insensitive, but again, here's another thing where this ontogeny, and we suspect that it's related to the timing of the spring transition. In the strong years, we get earlier upwelling. The fish may spawn earlier, or it might be the earlier spawn larvae that do better, but there does seem to be a pattern there with the earlier spawn fish doing better in the strong years. But again, there's, you know, the whole sweepstakes hypothesis. At some point, the later fish do okay, so you have to spawn over a range. That's another question we've never looked at. We've historically estimated the year effects above here in log scale as the main effects in a delta glim. We've also done a range of things, ANOVAs with comparable structure, and generally get about the same results with Julian day period and stations as fixed effects factors in the model. So here's the indices, and you can see there's a fair bit of covariation in there for the 10 different species. If you turn these into standardized anomalies and run a principal component analysis, what you see is that those 10 species are sharing a lot of, I mean, they're just all fluctuating in unison. The first principal component in that analysis explains almost 80% of the variation. The second explains a lot less. And the loadings are very even across all the species. So they're really, everything's kind of fluctuating in unison, despite there being some spatial differences in the distribution of the different species. So that's kind of interesting and kind of neat, and makes you think that there's some physical process driving the whole thing. And I was supposed to point out that here, again, 2013 was the largest, actually, index on record. So last year was the best recruitment we've ever seen. It was only a little better than 85, which was the second best by not a whole lot of difference. And various species themselves have had stronger years in the past, including Boccaccio, which really did the best in the early 80s when we did see strong recruitment. But in general, last year was really amazing. And having not been out at sea with Steve and Keith during the late 80s and early 90s when they were really abundant, you know, I first started going out here when things were dropping, and then here during the lowest years in the time series, and was kind of like, how could these things matter to the ecosystem? You go out when they're that abundant, and they're everywhere, and everything's eating them, and they're fat because they're eating lots of krill. And they really do represent a significant source of energy and trophic transfer to higher trophic level predators. They're the most abundant thing out there. So it was kind of amazing. And we've since heard from the power plant impingement surveys, divers, other things, that the last year really did seem to be. This is a picture off Point Sur from a diver. I forget what BOW stands for, but probably one of you divers know. This was online, but we asked for and received permission to post it. But it was just, these are mostly widow and yellowtail, I believe, Tom Leidig told me. But there's lots of your rockfish out there. So what's driving it? So Steve, again, had been looking at this for a long time, and it started, the first time he did this analysis, I think was in the late 90s, and he found it was sea level anomalies, relative sea level height, which has long been known from Shelton et al. Studies to relate to transport in the California current. When you have low relative sea level along the coast, it's generally an indication of strong southward transport. The California current is basically flowing very strong, bringing lots of subarctic water in, the gyre is kind of spinning, and the relative sea level is lower. So there's a lot of transport, lots of advection, lots of upwelling, lots of productivity, nutrients from the north, et cetera. So the index that he developed, we compared to ENSO, PDO, the Northern Oscillation, North Pacific Gyre Oscillation, sea level anomalies, and to a lesser extent upwelling anomalies had the strongest correlation. So we think that it really is an advection and a transport-driven process. When there's high transport, the rockfish and other eoic groundfish seem to do better. So we compared this then to the recruits from stock assessments. So we took 5 assessments that were age-structured assessments and had recruitment estimates. These are the 5 assessments for widow, short-belly, canary, chili pepper, and boccaccio. They're on log scale, again, so the fluctuations don't look quite as extreme. That's the only way the populations are at very different size. It's the only way you can get them on the same graph. Turn those into standardized anomalies. The shared variation is not as much, and this should be expected, right? They're settling out in the different habitats. There's different things driving, but they do still share about half the variation. The first PC explains almost half the variance. And again, you see that in some of these years, 84 and 99 in particular, they all sort of have strong year classes. So there's a lot of shared variation in year class strength. And it goes beyond these 5 species. If you look at like the 20 top groundfish species, most of those also had strong year classes in 84 and 99. Most had weak year classes in 98, 83, the El Nino years, 2005, 2006. So there's a lot of shared variation going on there. And the first PC of the indices from the core area correlates pretty well with the first PC from our survey. Not great, not as well as we'd like, but it correlates reasonably well. Again, the outlier is 99. The stock assessment common trend was very strong, positive recruitment in 99, and the trawl index was negative. So, and if you look at the individual species correlations, these are R's, not R squared in this graph, to the recruits, they weren't that great. The general threshold for people who've looked at recruitment indices is that you need to explain at least half the variance in order to make an impact on the assessment, to be informative for the assessment. And we weren't quite there for most of them. But we did do pretty well. We think we were doing well at indexing what was out there. During El Nino events, it was very low, not as strong El Nino in 5 and 6, but we know everything was crappy then. But this became a major problem then in moving forward, starting with that boccaccio assessment on how the boccaccio thing went down, and looking at some of the other stocks. It became apparent that everything was having a strong 99 year class, and we mixed it. So we were trying to figure out what the problem was, what to do. Looked at seabirds and salmon and other things that eat the juvenile rockfish. This is Kyra Mills did a paper with Seidemann, with Steve and Tom Leidig. I think they also compared to some of Tom's subtitle dive surveys. And they found, in fact, that the rockfish from this trawl survey were strongly correlated with a fraction of rockfish in seabird diets for murres, guillemots, rhinoceros auklets. These things eat a lot of rockfish when they're available. They tend to do better when rockfish are available. And so we felt confident we were indexing what was here. The question was, what were we getting wrong? So when in doubt, hold a workshop. That's an ancient government proverb, or mandated. So we held a workshop in 2006 and came up with a number of recommendations. I won't go through them in detail, but essentially the consensus was the spatial scale of the survey just isn't broad enough. I think we're missing it. If we have coastwide stock assessments for these species, we need either a coastwide or a broader scale survey to index the recruits for them. We also determined that the Whiting Co-op and our survey catch rates they were a little different, but they were comparable enough that we could kind of mix them. The change in abundance was orders of magnitude, and we were off by 5 or 10%, and we could account for that. We were looking ahead towards ROMS models and other things, helping us understand these mechanisms that would hopefully make it easier to come up with recruitment indices in the future if we could parameterize the models well. And we also recognize that the data were giving us good insights into ecosystem dynamics. So this gave us the green light to continue the expanded survey, to work with the Northwest Center. And the basic idea was that after a longer time period, we could go back and explore some coastwide data and get a better sense of whether coastwide indices perform better, which we hope they would. But then, budget crises hit, so we didn't manage to sample the northern part of the California current in 2010 or 12. We did in 11. I just don't have the numbers yet. I've actually gotten them. I haven't gotten them into the chart, though. And then in 13, we had very good coastwide coverage. This year, we should have very coastwide coverage. So the plan is to be able to rigorously then evaluate whether or not we can even do this, or whether there's something else going on driving this recruitment. We think we can. In general, we see a lot of coherence still when we develop these coastwide indices, and we compare the coastwide to the core area indices. These aren't the best comparisons necessarily because chili pepper, the biomass is centered in the core area. Same with short belly. Boccaccio, it's centered in the south, but I think we perform a little better when we have data from the south. We need to do this then with some of the other species from the north. But in general, we're not seeing huge differences between the core area and the coastwide area in most years. The exception is in 5 and 6, which were these unusual ocean conditions where we had, especially here in central California, really warm, nutrient-poor water piled up against the coast. There was no upwelling. We had Humboldt squid all over the place, eating everything in town. The salmon runs that migrated out of the San Francisco Bay into the Gulf of the Farallones that year were the worst. There were disasters. Those were the lowest runs that shut down the whole salmon fishery for several years. So these years were very anomalous, and what we found is that in most years since then, and we think before then, the core of the distribution of Yori rockfish is actually sort of close to central northern California, that we actually are in the core area, even though the core of the biomass is further north for more species. But in those years, except for chili pepper, everything was pushed either to the north for the Sebastoma species or the south Sebastodes species, the short-bellied Boccaccio and chili pepper. So we would have missed everything if we didn't have a coastwide data because there were fish in the north, and in the south, actually 2005 wasn't too bad for Boccaccio and a few other species. So this made us recognize that in most years, we probably do okay, but every few years, something like 99 or 5 and 6 comes along, and we do totally miss it. So we've embraced doing coastwide surveys now. I'm going to skim through some of the process studies. Keith did a nice paper with Steve and Eric Bjorkstad on fronts. Part of the story with missing 99 is that it was a huge upwelling year. Upwelling was very strong in May, especially that year. Frank Schwing and others did a paper on that. The water was a lot colder than the water we usually see. We often see higher catches of Sebastes at the fronts between about 9.5 and 11 degrees Celsius. The water was a lot colder that year. Upwelling was very strong. Water was essentially being blasted offshore. So we think in retrospect, if we'd gone 150 miles offshore, we would have found the rockfish, and later in the season they managed to make it back either to the south or the north. But in the central area, everything was just pushing all the water offshore. There's no krill, no nothing in the water. There's a lot of chlorophyll, a lot of upwelling, a lot of nutrients, but nothing else. So this is helping us try and figure out whether we can get away from the fixation effects and do something more along the lines of adaptive sampling. Eric Bjorkstad is working with Kat Crane to analyze Yoi rockfish otoliths and look at variability in growth in relation to environmental conditions, focusing on this early, that critical larval period between 5 and 50 days. What they found so far is that SSD correlates the best with growth, less so upwelling in some of the other indices. And she's very close to defending, but I've heard that for the last 6 months, so we'll see. Eric is using a lot of that data to parameterize these individual-based models, IBMs, and try and look at these mechanistic processes with a two-dimensional ROMS model to see if he can figure out, if you start on this day in this place, where do these particles end up when you account for some of the circulation patterns? And can you then predict when a rockfish is going to end up in good habitat, when it'll end up in poor habitat, and use that to sort of refine the indices. And what he's found is basically you're looking at the probability of being extruded, the probability of surviving over time or space, and then the probability then of contributing to recruitment. And by fitting some of these models, he actually was able to, let's see, in the top one here, he modeled the fit to the chili pepper index, and the bottom is what we had essentially without his model. So we missed 99 here, but with his model, he was actually saying, based on where the rockfish were, how big they are at that time, and how the ocean conditions played out after that, he could tell that 99 would be a strong year class. And what we're hoping to do is next move on to the three-dimensional ROMS models, things like Chris Edwards and other people at UCSC are developing these very good models. And if we can parameterize those, I think we'll start learning a lot more about this. So again, I was going to skim over that because I wanted to get to the ecosystem stuff. We catch a lot more than just rockfish. We're catching the hake, young of year Pacific sand dab, and again, the last couple of years, highest catch rates we've ever seen. Market squid, we get a lot of market squid, and we're really interested now that this is one of the most important fisheries in the state and trying to do more with this data. We do catch sardine and anchovies. We don't catch a whole lot of them. We don't think we catch them very well, but in fact, we think we're a decent reflection of at least their availability that early in the season, which is a little early for them. We usually only catch them when it's a weak upwelling year. When it's strong upwelling, they're either further offshore or further south. But in weak upwelling years, we seem to see them. And we also catch a lot of mesopelagic species, krill, and a number of other stuff. So in the last few years, we've had these sort of indicators in Cal Coffee reports and some other things as general sort of indices of how the Micronekton, how the forage base is doing. There's a lot of covariation among all these things, so we are doing these PC-based indices. And again, one of Steve's projects, now that he retired, was to kind of go through and do this really robustly with as many species as we can. So we looked at all the things we caught. We picked the 20 things that occurred in at least 10% of all the midwater trawls, different life history stages, all these different species. Some of them, krill and rockfish and dabs, we see an awful lot of. Others, deep sea smelts and sardines, are a little more rare, but we think we see enough, between 100 and 150 trawls a year to have a reasonable index of abundance. We ran this through, again, a PCA. The proportion variance explained by the first PC was a little low. What was interesting, and we played a lot around this, is if we only took like the top 8 or 10, or if we took all 30 to 35 that say show up in 5% of the trawls or more, the PCs, especially the first two, look identical. So it's really, there's a robust signal there that's being carried through. It gets a little noisier for the rarer species, and that explains a little less of the variance every time. But if you look at the PCs themselves, they don't really change. All the PC ones, whether we had 8 species or whether we had 35, essentially look like that. So we thought that was pretty interesting. But we settled on the 20 and tried to look, describe the PCs as an index of the relative forage availability in the system. The loadings sort of load the highest loadings on young of year rockfish, groundfish, sand dabs, hake, lingcod. Octopus is not actually a yoic groundfish, but it's kind of lumped in there anyway. Loligo, which should be Doryteuthis, I know, I'm sorry. Krill, this is Perictes, plainfin midshipman. We get quite a few of those, adult hake. And then there's the Clypeids and the mesopelagic species are loading on the other side of PC1 on that axis. So the dominant signal really differentiates between things that have high sort of annual productivity and things that have a slower life history and tend to be associated with warm, less productive, or less recently upwelled waters. As with the rockfish index, the best variable to explain this is relative sea level height. It relates very well to the species composition. So when you have relative low sea levels in San Francisco or coastwide, you tend to get more of the groundfish-dominated forage assemblage. When you have high sea levels, it's indication of less upwelling, less productivity, less advection and transport. That's when you get more Clypeids and mesopelagic species. And then PC2, I didn't throw a graph up. It seems to relate to the general trend. It seems to be kind of a linear trend for many of the indices, with explaining declines, say, in rockfish or some of the other groundfish, or increases in abundance for octopus and squid and other things. We're not entirely sure how to interpret that beyond that, but PC3 had a pretty reasonable correlation to the PDO. Interestingly, the correlation got stronger with longer lags, especially after a 1 to 2 year lag. The correlation got even better still. It actually got fairly tight, and we've been talking to Nate Mantua, who is now at the lab in Santa Cruz, and trying to scratch our heads and figure out if we can understand why exactly that is. But we don't, so in the paper, we just put the contemporaneous lag and explain it a bit more. One of the things that Brian is trying to do then is put these indicators together as part of the integrated ecosystem assessment, report to the Pacific Council, Cal Coffee reports, things like that. Try and explain and a snapshot what's happening in the ecosystem. See if we can do that. So what this is, and this, I think the first time I showed it was Western groundfish. This is the second time, so it's still very experimental. These are essentially stacked anomalies. So the standardized anomalies, but all stacked together and kind of color-coded relative to the relative weight on PC1 with PC1 itself superimposed on it. So the warmer species, again, sardine, anchovy, krill, and myctophids are sort of the lighter colors, and the bluer colors relate to the Yoi groundfish and things like that. And we think this is a reasonable way to kind of portray at least what's happening with this Micronekton assemblage in May, June. Things often change quite a bit during the season, but in May, June, this is kind of where, which is when salmon migrate out, when seabirds breed. This is kind of what the system looks like. And so we think it's useful, but feel free to shoot it down later when I ask you to, for questions. But again, 2013 was just off the charts. It was a crazy, a lot of transport, a lot of productivity year, and that's really reflected here. I don't think I mentioned, by the way, that they didn't enumerate all these things starting in 1980, so the reason the sort of community assemblage starts in 1990 is that's when Steve Ralston came on board and was like, you're not counting this stuff? You're just guessing? You got a subsample, and like, what's going on here, guys? So we have some data prior to 1990, and for some species we think it's reliable. For most of the others, we're not so sure. So for most of the community stuff, we start in 1990. So that's some of the temporal stuff. Jared Santoro has been started doing a lot of the spatial analysis of the Micronekton, trying to establish these climatologies. Where do you see the different organisms, and why do you see them there? So looking at the relative catch rates, where red are higher catch rates, blue are lower catch rates for many of the different species. He did that for much more than these. And then working with Isaac, he sort of did these climatologies. This is, again, over a 20-year time period of mean physical ocean conditions, so wind speed, salinity, stratification, temperature, peak decline depth. I know chlorophyll isn't a physical condition, but look, just cut us a little slack. So looking at mean chlorophyll values. And the idea was to link those together. So do some spatial PCAs to understand how the environmental seascape related, how those different variables related to one another. Do the same thing with the biological seascape, with the spatial climatology, and not surprisingly, what he found is that the physical habitat is absolutely driving the spatial structure of the Micronekton. So you have things that are only over the shelf, things that are only offshore, things that straddle both and are evenly distributed. And it was, I think, a nice, elegant analysis, and paved the way to start looking at the temporal variability then in those processes. And then he did some cluster analysis to try and better understand sort of what mix of species you see in what places and why that is. So again, the environmental clusters lined up fairly well with the Micronekton clusters. He overlaid some of the higher trophic level data from the seabird and mammal observations on top of that. Again, things look a little different because we're taking these data continuously while we're transiting instead of taking the data at fixed stations. But using GIS, you can obviously mix and match things together to understand how these things map. And what he ended up putting together was this characterization of different ecologically important areas, sort of where you had sort of a general sort of suite of animals and where you had finer scale differences in the community assemblage, and why that was mapped entirely to the physics. So that was nice. More recently then, we've been trying to work with, follow up on some of the work with seabird breeding success and productivity as related to the availability of Micronekton. So there's an increasing interest in the relationship between forage species, how they're managed, and the productivity of higher trophic level predators, especially seabirds. Philip Currie and a bunch of co-authors had a really nice summary of that, relating sort of forage availability to the breeding success of seabirds. I think it was called 1/3 for the birds. In the California current, this is basically bird breeding success mapped onto rockfish. And again, you saw how variable rockfish are. They fluctuate by several orders of magnitude, even for the unfished species. So where's a third of what is what we were wondering. So we launched on an analysis to try and see if we could detect what the actual effects of fishing on adult rockfish were to the availability of juveniles. So again, in Central California, murres, auklets, and pigeon guillemots have long known to, they feed primarily on rockfish. They tend to do better when rockfish are abundant. So we related their productivity to the abundance of juvenile rockfish. Not perfect relationships, but significant and pretty reasonable. Then what we did is try and figure out how many juvenile rockfish would there have been in the absence of fishing. So we ran stock assessment. We reran all the stock assessment models for these species, removing the effect of fishing, basically like fixing their parameters and running the model again without any catches. So what you see is what we had for the base model and the base stock assessment, and what we got for that model. And you get a little bit of variability. It's not as great as we thought, but it's more variable for some species than others. For Boccaccio, it was actually kind of interesting. It looks like, and I do believe this given how sensitive they are to climate, how variable recruitment is, Boccaccio would have declined naturally to about half of their previous abundance in the absence of any fishing at all, just because the environment was so poor in the late 80s and early 90s. Short-belly, of course, are unfished, so the lines are almost on top of each other. There were a few catches historically. So basically what we did then is scale the abundance of juvenile rockfish up relative to these differences, assuming, again, that because it's density-independent mortality is what the main driver at that point, that relative abundance of pelagic yoi at that time would have scaled to the relative difference between the fish and the unfished species. And by doing that, we could then use that the fitted relationship to predict what the breeding success would have been in the absence of fishing. And in general, it wouldn't have been that different. Most things would have done a little bit better in most years. Pigeon guillemots were the most sensitive because they have a different reproductive strategy. They'll produce multiple clutches or multiple broods, and so they're more sensitive to finer scale differences in the abundance of prey. But common murre and rhino otoliths have a little more conservative strategies, so they generally, the difference wouldn't have been more than, say, 10 or 15% greater productivity. But for the pigeon guillemots, there's some years where they would have had quite a bit more productivity had we not been fishing. But in general, this gave us something that could kind of balance the create-all analysis on. What it actually also did was give the Pacific Fishery Management Council a rationale for setting basically no fishing or trace amount of fishing on short-belly rockfish. Short-belly rockfish has never been fished, but they're one of the most abundant species on the West Coast, and there's long been concern by the bird and mammal people that if you start fishing this thing, the birds and mammals will suffer. But we've never, it's always been on the books as sort of a 10,000 metric ton catch because no one's really looked at it. So there's a lot of it, so you could probably catch 10,000 tons. And we started playing around too with some a little dicier, but sort of what happens if you didn't fish short belly down, what happens if you overfished it, what would have happened to the birds then, and the impact would have been quite a bit higher, and that was enough to convince the council that let's kind of leave this one alone, at least for now, until we know a little more. They took the total allowable catch down from, I think it was 13,000 metric tons down to 50. So, and that was considered, you know, that was part of Oceania's effort to conserve forage and a bunch of other people trying to, you know, get the council to do more ecosystem-y things. But we like that as one small success in moving in that direction. So in following up to that, Jared started looking at the sort of spatial patterns, relative abundance, and spatial patterns in abundance for a lot of these species that the birds like in particular. So they eat a lot of dabs, juvenile rockfish. And if you look at sort of Monterey Bay, the oceanic region, and the shelf region, what you see is that the yoi groundfish are essentially very widely distributed, and they fluctuate in phase throughout all these areas. By contrast, krill and anchovy don't. There's very different signals over the shelf than you have offshore, and basically krill in particular are much more variable in the northern shelf in the Gulf of the Farallones than they are in offshore waters or even in Monterey Bay. And anchovy are a little bit the same. And so our goal then was to try and understand, okay, if these species matter to the birds, where do they matter? And can we get a better understanding of what areas are the most important for some of these things? So he started looking at, again, relating these PCs of physical conditions to the relative abundance of krill, rockfish, dabs, and anchovies over time, and seeing where the physical conditions seem to drive the relative abundance. From there, he moved on to looking at the krill, rockfish, and sand dabs as the primary forage for the seabirds and relating those to the seabird productivity in different spaces. Not surprisingly, the Farallon Islands regions consistently falls out as, you know, the place where krill, rockfish, and sand dabs correlate best to seabird productivity. And from that, he did a set of GAMS, generalized additive models, to again revisit this mapping of the abundance of forage to seabird productivity, but do it at the right spatial scale and try and understand then where do these things matter, not just what matters, but where does it matter. And so he's actually had, you know, I think some of these GAMs might have been a little too loose, but in the most part, you see essentially the same kind of relationship that Creole and others found with respect to the relationship between some of the key prey items and the seabird reproductive success. So that's nice. We've done sort of similar work with Pacific salmon. Again, we're indexing a lot of the same things that salmon eat. Julie Thayer just did a paper where we compared salmon diet data from the early 1980s with some data she collected in 2005 through 2007, again, those warm, poor, low productivity years, and one paper by Merkel back in 1955. And what we found is relating the diet to the trawl survey did pretty well. We were, again, seeing what the salmon saw. If you look at Simpson's diversity index of what the salmon were eating, the diversity has been going down over time. There's basically been a less diverse assemblage of forage available to them. I think this would have gone back up if she'd kept it up, but unfortunately she ended during that low productivity regime. But it does speak to the fact that, you know, it's not just one or the other forage, that it's this suite of forage species, and that what she looked at a little more closely was the timing, that many of these different things sort of came online and were available to adult salmon at least at different times of the year, with herring coming early, and then krill, and then rockfish, and then other ground fish, and then anchovy and sardines. So, you know, these predators really need a suite of forage available to them. And then Brian Wells has been doing quite a bit of work. Again, this is a trajectory of the Sacramento Index, so Central Valley salmon. The blue line is the model that the council uses based on the sibling model of Jack returns the previous year to estimate to forecast what that year salmon return will be. And Brian developed a different model based only on krill from the survey and relative sea level, and basically does as well, if not better, and you'll get different answers about who's better depending on who you ask, but definitely very comparable to the council model, based, you know, ignoring the siblings and just focusing on sea level and krill. And so, and he's been basically getting the same kind of forecast in recent years that the council's been getting. And they've taken a look at it. There's a lot of people interested. The appeal is that it's a nice mechanistic relationship. There's some drawbacks too that the salmon people have raised, but for now, it's, you know, it's something that's ongoing. I don't know if Valerie made it, but she'd be mad if I didn't show. Oh, you're right here. Hi, these are for you. So one of the things that we do see a lot of, and we've also neglected to do much work in over time, is gelatinous plankton, so jellyfish as well as salps and other things. Over the last couple of years, I tried to put together at least some numbers. One of the challenges here is that what jellies really initially brought us was nothing but grief, because we would have these blowouts. We'd blow out the net when we set in jellyfish territory, to the extent that we had to drop a lot of the coastal stations and add some offshore stations to account for, you know, these jelly blowouts. That would, if you set on a bunch of jellyfish, you blow out the net, and then you spend the whole day trying to sew it back together so you can fish again the next night. So jellyfish cause us grief, and as a result, then we couldn't really use the catch rate data we have without accounting for the blowouts somehow. So without getting into details, we took a stab at accounting for the blowouts and developed these jellyfish index. We've for Aurelia and Chrysaora, this is what bleeding a blowout that didn't actually break the net looks like for the salmon folks. But when you get a lot of jellies, you get a lot of jellies. They're an important part of the ecosystem, an important player, and so we were trying to understand a little more about them. When we started mapping out the climatology, the spatial distribution, what we also saw was that the spatial distribution overlaps very strongly with Thysanodontis spinifera and juvenile salmon, suggesting then that there's some kind of interaction there. Both the jellyfish and the salmon are preying probably on different life history stages of the same thing, but it would make sense then that there's something going on. Rick Broder and John Rizuka up in Newport have been looking at this quite a bit as well. They're a little ahead of us in terms of modeling, but we have the data to start moving ahead and understanding this a little better. And then we also catch salps. We caught a lot of salps in 2012. It was just absolutely surreal. It was a different universe. It was a gelatinous universe out there. So it was a low year for the scyphosome, but huge year for thalassians, doliolids, and heteropods. So you got the pyrosomes. And then we were doing a little bit of hook and line fishing to see, well, what's the response? And yellowtail rockfish, especially, and some of the other rockfish we caught, were just gorged on salps. I mean, there can't be a lot of caloric value, but anything's going to give you some caloric value if you eat enough of it. And this one had at least 100 salps in its stomach. So they really dominated the ecosystem. But what they really did was hang up on the net and cause us to blow out the net repeatedly. We had, until that time, here's a number, the fraction of toes that had been in jellyfish blowouts. So we switched these stations out around 1990, dropped some of the worst stations and added some different ones, and the blowout rate went down, went back up again in later years. We'd never had a blowout due to salps until 2012, and that year we had 8. 8 times we ripped the net to shreds. Once we twisted the bridles, ruined all the wire. It was just a nightmare. And at the same time, the SALPs were, knocking Diablo Canyon offline and getting a little bit of news attention. So they were absolutely dominant. And they actually enumerated, SALPs were one of the things they did enumerate reasonably well starting around 87. They dropped it in 2001 because we had too much other stuff to do. The first week into 2012, I said, let's start counting them again, because for crying out loud, look at how many there are. They actually matter. And again, these are log transform catch rates, so you can see that orders of magnitude greater abundance in 2012, and then still fairly high in 2013, especially in the south. So another data set just waiting for a grad student. I'm going to skip the summary because I'm getting tired of talking and it's almost 5 o'clock, and just acknowledge, in addition to all the people I listed, like there's literally been hundreds of captains, officers, crew on the Star Jordan and other ships, the scientists, volunteers, we had teachers at sea, lots of grad students, undergrads, people like that who come and have made this all possible, including the taxpayers. So thanks. Greg.
I'm actually a little surprised that the PDO factor didn't weigh more heavily. You compare it to the Francisco Chavez summary paper that a lot of other people's work. And he made a big deal out of the 3 PDOs in the last century.
Yeah.
Is that breaking down, or is it just that they're looking more at physical data like sea level changes, and those just happen to be contributing?
Yeah, well, there are, now the MPGO is a hot thing, and it started with sort of the second mode of the PDO. The thorium mode was becoming stronger than the first mode, so that might have a lot to do with it. We did look at both modes of the PDO, and neither of them did that well. Of course, there was something to it. My recollection is that the PC1 for the community index also loaded reasonably well on the PDO with a 2-year lag. But we really don't understand why the lag, why should there be a lag like that? It is a Gulf signal, and maybe it takes that long to transmit down here, but these are animals, which are 100 days old, so why are they responding 2 years later? So the weird lags have a lot to do with it, and I think we think there's more to it than we've been able to tease apart so far, which is why we're trying to corner Nate in the hallway every time we see him. Explaining why this happens, Nate.
Right, there's certainly got to be a species composition part of that. I didn't see Spinobrachys in there, but I saw Tarleton Venia, and I didn't,
I think Spinobrachys is in there.
I didn't see it listed. The point is that those species can tell you whether there's a water basin down south or up north, it's here or inshore or offshore. And mesopelagics can tell you that. Oh,
they tell us a lot. And I've talked to Tony Coswell a lot about this, and in fact, our index relates very well to his index. And it's interesting, like our PC3 is strongly correlated with his PC1, which is the one that he relates to PDO. And then he's got the other index that relates to the OML and depth of the picnicline and some of the others, and that relates well to our PC1. So I think part of it is that we're sampling sort of a different mix and getting a slightly different signal than he is, but we're seeing kind of the same things for mostly understandable reasons. So we've exchanged some time series and some correlations and some notes, and he's back in Australia, so I can't pick on him anymore in the near term, but we are planning to get together. What I plan to do is do some of these indices now for the south and the north. They'll be shorter, but we can at least relate them to his other indices and see if we get a better correlation in the south, or see if we can understand why it's taking so long to pick up that signal.
So let me just add on to that for a second. Francisco is also saying that these signals are not as distinct as he once thought they were. They don't mean the Doki, and there are correlations with other things as well. So it seems to me that the atmospheric dynamics are more subtle than we thought too.
And that's why it's looking more and more like the MPGO, at least in recent years, is a better index, although that's not helping us explain much more of this. But I think that part of it is also the local processes. So 2005, 2006 were very different in Central California than they were in the Southern California Bight or in the northern part of the California current. And so these regional processes are important enough, at least, to start some of the larger signals from the PDO, I think.
Yeah, my working with various indices from the Southern Ocean is the fact that there are more there are more atmospheric oceanic coupled processes
Yeah.
going on that you know
Yeah.
about. And the permutations, the combinations, and the variations of those over a 100-year period, we're never going to get it right. You know, I mean, it's, you know,
We'll get better, but yeah.
just when people think they've got it,
It breaks
all of a
down.
sudden
I
it
know,
breaks
always.
down and they go, oh my gosh,
Well, that's, yeah.
things aren't the way they were supposed to work.
Yeah, there's a lot of these things are nonlinear and interact in very strange ways. But that's one argument we've made for why it makes sense to monitor sort of what's happening, you know, in the middle of the food web. You know, get above the physics and the lower trophic level productivity and see what's happening in the middle, because that's what most of the higher trophic level predators are seeing. And even if we don't understand everything about why things are booming or busting in the middle, at least then we'll understand that things are booming or busting, and we can better understand then
And
how the
this-
top higher trophic level predators are responding to that shift in the availability of prey.
The southern ocean's the same way, there's a whole suite of species
Yeah.
that are doing well under a certain
Mhm.
suite of environmental
Yes.
conditions.
Get there.
My question, and Val maybe sort of answered this question, was, is there any good way to predict, how future climate conditions and things might affect recruitment of rockfish along the coast in terms of changes in the intensity and the timing of upwelling and stuff like that, from some of these surveys that you guys have?
I
Or
hope
is it
so. I mean, I think
impossible?
no, I think some of what we're hoping to do is really move forward. So I didn't talk about this either, that Brian and Jared have been working with Chris Edwards and others with their 3D ROMS model at UCSC, calibrating it essentially to krill and seeing how well, if you calibrate their model, it starts performing better, you can compare the observed krill distributions to what they predict. And our hope is next to start doing that with rockfish and some of the other things. And then once they're at a stage where they can run their model forward, based on some of the climate model projections, we should be able to get a better idea, what does the future hold for rockfish recruitment. It might actually be promising, because if we get, stronger backing things, stronger wind gradient, leads to more upwelling, leads to higher productivity along the coast, we might do better, the rockfish might do better, so it might tank. But it's nothing else, so you run it through a model and then guess and go through it.
John, so there seems to be every once in a while we come up with a new environmental factor that we can measure. We get CODAR information relatively recently. All these things. If there was do you think we're missing an environmental physical factor that we're not measuring very well? That you think would be the next, if you were putting billions of dollars into sort of monitoring something else, besides, we use satellite oceanography, which we got that's.
I think oxygen is a big one that we're not getting well. I think they're starting with this little army of gliders that are out there, but we have some decent baseline data, but I think oxygen, especially looking towards the future, is really going to be bright. I mean, some of the work we've been doing with Humboldt Squid, with Bill Gilly and others, is really showing that a lot of that invasion was probably driven by shoaling of the OML. Oxygen, the shoaling of the OML is going to displace some of the deeper rockfish, maybe some of the other deeper fish, and start shifting habitat around. These little low pulses might relate to oligo productivity styles. So I think oxygen is the one that we're probably not doing too badly, but the one we should really be paying attention to looking in the future, as far as I can tell. Other than that, I mean, there's a lot of data. I mean, you talk to the ERD guy every time we get Isaac up, like, what can we use instead of coastal sea level? We'll be like, well, here's the Ibiza data. You can get all that satellite data, telemetry data over the last 20 years. I think it starts around 1990 or so. So he's gotten that to us, and we're starting to use that instead of relative sea level from the tide gauges. It, not surprisingly, it gives you about the same signal, but it's probably a little more robust. Yes?
Do you think this year's gonna be a good recruiting year for rockfish?
I would guess so. I mean, you've heard tale of El Nino coming, but it hasn't come yet, and the signs would be pointed to it. I think one of the things that Eric Bjerg mentioned is that we had this long spell of really calm weather in January last year that probably related to that critical period. Might have been the thing that gave a lot of the recently extruded larvae just the boost they need to do well. And I think we sort of had something comparable in January this year, where didn't get a lot of storm systems, didn't get a lot of rain, probably developed some nice stratification that might have been enough to lead to strong recruitment. Ask me again, and yeah, with some upwelling and some wind now, exactly, a little later in the season. So I'll tell you in May.
Okay, well, I think that's will do. Thanks.
Okay.